The fundamental part of gene bank management is not only to capture genetic variation from the field, but also to maintain that variation over time, because we want to treat these accessions as reagents, essentially, as scientific reagents. And so we don't want them to change substantially over the course of their ex situ conservation. The difficulty with wild material is that they are highly diverse. So within a particular accession or a unit, there might be lots and lots of genotypes. And there are lots of genes with different gene frequencies that compose those genotypes. And so every time that you regenerate that material, it's a sampling event that may, in fact, lose the rarest alleles during the course of regeneration. And this is sort of a fundamental issue in population genetics and sampling dynamics. So you could do this and simulate it. You can use jelly beans in a jar and take repeated samples. But the process is more difficult with wild material because they're much more heterogeneous. And retaining that variation requires at least some thought about the size of the sample that you want to take and the structure of the seeds that you collect. For instance, many of our samples get distributed over the course of their time in the gene bank, and they get down to a point where they need regeneration. A lot of logistics is then brought to bear on how to do the regeneration properly. For cereal crops that are much more uniform, the difficulty is fairly trivial. Individuals are then pulled from seeds that are from that remaining sample, and they're regenerated in a standard 1 by 2 meter plot, probably around 100 individuals. But for wild materials that are really diverse, one might expect to have a huge sampling event if you only had 100 individuals. And so from simulation studies, we can see that upwards of 1,000 individuals are warranted, where you're taking seed from much more diverse representation of that accession. The other part of maintenance and retaining that diversity comes from checking the quality and the diversity of that seed incrementally over the course of regeneration. And there's only really 2 or 3 routine assays that are done on samples. One is a fingerprint analysis that's done with microsatellite DNA markers. And the Rice collection in the US has a routine checkpoint genotyping. That is, they check every time they do a regeneration, they check the initial accession, and then they check the derived seed from that accession and make sure that it's very similar. In most of our collection, though, we don't have any such sort of checkpoint genotyping. And so the curator is left with making decisions about the size of the sample based on the diversity of the material.